Okay, welcome. This is Chapter 5 review. This is the chapter on cells, the working unit of life. We're going to spend some time on this review looking at surface area to volume ratios of cells, microscopy, and also an intro to the basic cell types. So this chapter is looking at cells. Remember, we talked previously about this concept called the cell theory. And the cell theory very basically states that all life is made out of cells. This is the cell theory. Every time we find a new living organism, it's made out of cells. So it's been proven time and time again, and that's what a theory is. So all life is made out of cells. These are the fundamental building blocks of life. Okay, all cells come from pre-existing cells. And if you look at the bottom here, most cells are small. In fact, they're so small that we need microscopes to visualize them. So the question then is, why are cells small? Why are most life, the fundamental building blocks of cells, small? And another way to think of it is, why are humans made out of trillions of cells instead of millions of bigger cells? Or why are they made out of trillions of really small cells instead of hundreds of thousands of big cells? And the reason for that is because we have this concept that's called the surface area to volume ratio. The surface area to volume ratio. Okay. So what do we mean by this? Okay, the surface area is the coverage around a cell, so the plasma membrane. Okay, the volume is the amount of space in substance inside of a cell. So how do these two things relate to each other? Well, if we look at the volume, what the volume does, okay, the volume, it determines the amount of chemical activity inside the cell at any one time. So if you think about inside a cell, stuff has to move from one side of the cell to the other side of the cell. The bigger that cell is, the longer it's going to take. So if it's important nutrients, how do they get from one side to the other? If it's waste, how does it move from one spot to out of the cell? Okay, it moves through a process called diffusion. Lots of things move by diffusion. It's a random movement. There's also active movement. We'll learn about this later in the chapter. If you think about if you're in a large room and somebody has a cup of coffee, it's going to take a long time for that smell to get from the front of the room all the way to the back of the room. That's diffusion. But if you were in a small little closet and somebody had a cup of coffee, everybody would be able to smell it very quickly. So this is the limitations. If cells get too big, they're not as efficient at having things move around inside of the cell. So that's part of the equation here. The other part is the surface area. So the surface area to volume. The surface area determines how much stuff can come in and out of the cell, because the surface area is the coverage of the cell, and that's the plasma membrane. So as a cell gets bigger, the surface area gets bigger, allows more stuff to come in and out. But the problem then is that as the cell gets bigger, it determines how much stuff comes in and out, but the cell's getting bigger. So once that stuff gets inside the cell, it's going to take longer for it to get around to where it needs to go. So there has to be some sort of happy medium here. And this brings us to the concept of the surface area to volume ratio. And it turns out it's most efficient for smaller cells because they have enough surface area to bring the stuff in and out they need, food, oxygen, for instance, but they're small enough that the stuff, once it gets in the cell, can diffuse around in a reasonable amount of time. Okay, let's look at some examples here. These are, at the top, these represent different sizes of cells. We have a small, we have a medium, a large. Okay, these are all actually pretty small, 3 microns. It's a very small diameter. And so we go across here, increasing cell size. So as these cells get bigger, not only does their surface area increase, okay, the surface area increases right here from 3.14 microns squared, that's squares are the units for surface area, all the way to 28.26 microns. So it gets bigger as a cell gets bigger. That makes sense. The volume also gets bigger, but what we're concerned about is the ratio. So the surface area to volume ratio is what we're really concerned about right here, surface area to volume ratio. So for the small cells, this ratio is 6:1. Okay, so it has a high surface area compared to its volume. So that means it has a big surface area, allows lots of stuff to come in and out of the cell, but its volume is still small, being only 1, that allows stuff to move around inside of the cell. So it's pretty efficient, big enough to allow lots of stuff to come in and out through the membrane, small enough that it allows stuff to get around inside of the cell. Let's look at the bigger cell. The surface area to volume ratio is 2:1. So the surface area is only 2 times as big as the volume. If you look at the surface area, it's about 30 microns squared. The volume's about 15. So it's about, the surface area is about twice as much as the volume. So it's a 2:1 ratio. So here it has just about twice as much surface area to allow stuff in and out compared to the volume inside. All right, so you get the idea that the surface area determines how much stuff comes in and out, the volume determines how much that stuff moves around inside the cell. Now don't get confused that there's some exact size that works best for all cells. Some cells are bigger, some cells are smaller, but it's all determined based on their function. Okay, if they need a lot of surface area because they're exchanging lots of substances, they can be smaller. Okay, some cells get bigger, but they're not going to be as efficient at that. And it depends on their environment that they find themselves in. So this concept of surface area is another one of those golden rules in biology that we've learned about so far. Oftentimes, cells, structures inside of cells, and organisms themselves will make adaptations to increase their surface area to interact with their environment. Okay, so that's a major issue or an important concept when we're talking about cells. All right, so we're mentioning cells are so small and then you need a microscope to see them. There's different types of microscopes, right? The general type that you'll use inside the lab is called a light microscope. This is the type that sits on the lab bench and it has a light source at the bottom and it shines light through your sample and you observe it. The light microscopes are good for observing things at the cellular level, even some small subcellular structures. Okay, and these are some basic pictures of individual cells underneath a microscope. Okay, now light microscopes have a limitation, and that's that they can't, their resolution isn't so high, so you need a stronger microscope to see even smaller structures within a cell, and also fine structures on the outside of a cell. So we use a special type of microscope called an electron microscope. All right, this is a very special microscope. It requires lots of preparation of your sample. If you look at this picture, this is what it looks like. It's not the classic type of microscope that sits on top of the desk. You prepare your sample, you put it inside the chamber, and you visualize it on computer screens. And with this, you can have very, very high levels of magnification. Okay, and there's two main types. There's transmission electron microscope or microscopy. This is called TEM for transmission electron microscopy. And what this does is it takes a cross section of a cell, so it allows you to see inside. So here's a cell that's been cut in half, and we're looking inside of it. Okay, so you can see structures within the cell. Also note the scale bar, 10 microns. We have a very high magnification here. On the right, this is scanning electron microscopy. When you prepare your samples for this, the cells are not cut in half, and you can actually see the surface structure. So here's whole entire cells. Here's a cell. Okay, here's another cell. And you see all these structures on their surface. So SEM refers to the cells intact. So each of these, TEM and SEM, have their own general purposes and usefulness. It may seem like additional detail here to know about electron microscopes or TEM versus SEM, but it helps you orientate yourself when you see one of these pictures. Okay, these pictures, if you see one, they're called micrographs. These are pictures from a microscope. And if you know if it's in black and white, has a very high magnification, you can assume or likely predict that it's an electron micrograph. And so the thing you're looking at is very small, so you can help gauge what you're looking at. Also, if you recognize it's a cross-section, then you know it's a transmission electron microscope, or if it's a whole cell, it's SEM. In addition, this will be useful when you're looking, if you take microbiology, because you'll see lots of pictures using an electron microscope, because bacteria and viruses and other microorganisms are very small, and you need very high-powered microscopes to see them. Okay. Now we'll start kind of easing into looking at cells more specifically. When we want to look at cells, there's certain things that all cells have in common. Okay, even though there's different classes of life, they're all made out of cells, and there's basic functions of cells that are similar across all life forms. One of the basic fundamental properties of cells is that they have a plasma membrane, right? This phospholipid bilayer that surrounds it. All cells have a membrane because you need something to cover the cell. Now the membrane is so important, it has its own chapter we'll see in the next chapter. But some basic properties of the plasma membrane is, one, is that it's a selectively permeable barrier. Based on its chemistry, based on the fact that it's made out of phospholipids, it's going to interact with substances very specifically. Certain things come in, certain things go out based on their chemistry, so it's selective. Not everything can pass through it. And it's permeable, which means things can pass through it, okay, but only certain substances. The membrane is important because it also helps the cell maintain a constant environment. Okay, it helps maintain the water inside, helps maintain nutrients and oxygen and waste materials. The cell is important. It plasma membrane is important because it allows for communication and receiving signals. So if you look at this picture, there's some structures in the plasma membrane, proteins, that serve as receptors to receive signals. We also release signals from the membrane to communicate with other cells in our bodies. And then finally, the membrane helps the cells bind and interact with adjacent cells. So here's one plasma membrane in this picture. It's attached to a neighboring cell through this structure right here. So it allows connecting. So you need cells to connect to each other to form more complex structures. Remember, cells group together to form tissues, and tissues form organs. So how do these cells clump together? The interactions between cells and their plasma membranes and the extracellular matrix that we'll learn about later. And then finally, there are two main classes of cells we've learned about. Okay, there are prokaryotes, which lack a true nucleus in membrane-bound compartments or organelles. These include the bacteria. Okay, and then there are eukaryotes. Eukaryotes have a true nucleus and membrane-bound organelles. And these are things like animals, plants, insects, fungi, protists, which can be single-celled or multicellular, and fungi, which can also be single-celled or multicellular. Right? So the eukaryotes are a very diverse class, and they represent most organisms that you're familiar with. And prokaryotes are going to be the ones we'll focus mostly on, are bacteria, which have lots of important roles in the environment, even though they get kind of a bad rap for being pathogens. So bacteria, actually, most of them are good for us. If you look at these pictures, here's an example of a general bacteria, kind of a rod-shaped, not many defined structures inside. On the bottom, this is a eukaryote, okay, the plasma membrane around the perimeter. And then if you look closely inside, there's a nucleus, and then there's smaller structures inside called organelles. This next slide here, this really helps point out the main differences. Okay, so on the left are prokaryotes, on the right are eukaryotes. Prokaryotes have lots of structures in common with eukaryotes, actually. They have a plasma membrane, they have cytoplasm, they have genetic information. Okay, same with eukaryotes. But what's different is you lack a nucleus inside. Right? So for a prokaryote, the basic things you have are a nucleoid. This is a dense region where the DNA is found. You have ribosomes, which make proteins. You have cytoplasm, which is the water, primarily waters the liquid portion inside of the cell. Okay, then you have a plasma membrane. Outside the plasma membrane, different types of prokaryotes can have different structures. So they all have at least a plasma membrane, but some have a cell wall. The cell wall is made out of peptidoglycan. What type of molecule is peptidoglycan? Peptide means protein, glycan means sugar. So peptidoglycan is this mesh, kind of like a chain link fence of sugars linked together by proteins called peptidoglycan. And then some can have a second plasma membrane. Okay, outside of the second plasma membrane, some can have a capsule, which is an additional coverage. So all these features outside the plasma membrane, a cell wall, a capsule, provide additional levels of support. They also help us classify bacteria. So even though we say prokaryotes lack a nucleus, lack organelles, they still have important structures inside that you need to be familiar with. Okay. On the right, we have the eukaryotic cells, and if you look at the picture, you can tell they're a lot more complex. They have all these internal membrane-bound compartments, these organelles, they have a nucleus, but they still have a membrane, they have cytoplasm. And we'll spend some time looking at these structures in the next part of this review. So this is a general introduction. We got the basic ideas of surface area to volume, microscopes, and a general introduction to the plasma membrane and the two major classes of cells.